Hello, I'm back. I'm Siti Nurhana binti Makna with metric number of 207193. So now, in this video, I'd like to show how to prepare a bacterial smear and how to stain our bacterial smear with a gram staining. Before that, let me give an introduction to gram staining. What's that? Gram staining is the most crucial and most used differential staining in microbiology. This test differentiates the bacteria into gram-positive and gram-negative bacteria, which helps in classification and differentiations of microorganisms. Before that, the bacterial smears must be prepared in order to execution of any staining techniques such as gram staining. Although it is not difficult, the preparation requires adequate care. So here is the procedure of preparing of bacterial smear and also the gram staining.
Firstly, make sure the slide that you are going to use is cleaned. Then place it on a dry tissue. In this experiment, I'm using a solid culture sample. By using a proper aseptic technique, take one loopful of cell suspension from the jar and place it onto a clean slide. It is crucial to avoid thick or dense smears when taking the culture onto the slide. After that, take one loopful of distilled water and place it onto the cultures on the slide. With a circular motion of the loop, spread the suspension mixture into a thin area. It's actually good if the smear appears as a thin whitish layer of film. Then, allow the smear to air dry. This may be done by placing the slide onto the dry tissue or weaving the slide. Once the smear has dried, quickly pass the slide through the outer portion of this flame 2 to 3 times rapidly. By using the bacterial smear that has been prepared, place the slide onto a staining tray carefully. The first primary stain is crystal violet. Gently flood the smear with crystal violet and let it stand for 1 minute. After 1 minute, gently wash with tap water. Next is the grams iodine mordant. Flood the smears with grams iodine and again let it stand for 1 minute. Gently wash with tap water. After that, decolorize the stain with decolorizing agent, which is 95% ethyl alcohol. Add the reagent drop by drop until the alcohol runs almost clear, but do not over decolorize. Again, gently wash with tap water. The final reagent that is being used is safranin as counterstain. So, counterstain the smear with safranin for 45 seconds, then gently wash with tap water. And lastly, blot dry with a tissue paper or just let it dry.
So here is the expected result. When the bacteria is stained with primary stain crystal violet and fixed with the mordant, some of the bacteria are able to retain the primary stain and some are decolorized by alcohol. The cell walls of gram-positive bacteria have a thick layer of protein-sugar complex, we call it as peptidoglycan, and lipid content is low. Decolorizing cell causes this thick cell wall to dehydrate and shrink, which closes the pore in the cell wall and prevents the stain from existing in the cell. So, the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram-positive bacteria and appears blue or purple in color. But in case of gram-negative bacteria, cell walls also takes up the crystal violet iodine complex, but due to the thin layer of peptidoglycan and thick outer layer which is formed of lipids, the crystal violet iodine complex gets washed off. When they are exposed to the alcohol, the decolorizer dissolves the lipids in the cell walls, which allows the crystal violet iodine complex to leach out of the cells. Then, when again stained with safranin, they take the stain and appear red in color. So that's all from me. Thank you.